My name is Dr. Matute, University of Arkansas, PTC. This is an overview of blood. Blood is part of our internal transport system. Our internal transport system is normally referred to as the cardiovascular system or the circulatory system. When we refer to it as the cardiovascular system, we are describing it from an anatomical perspective. When we refer to it as the circulatory system, we are describing it from a physiological perspective. Now, what are the requirements for having a circulatory system? Number one, you must have a pumping organ, for example, the heart. You must have circulatory vessels, for example, veins and arteries. And then you must have a circulatory medium itself, in this case, blood. Now let's look at blood in details. Blood is a specialized connective tissue, and from a composition perspective, blood is divided into formed element and plasma. The formed element, they are the living components of blood, and plasma is the non-living component of blood. If we take blood and we centrifuge it, centrifuge means to spin, so we'll put it in a small tube, and then we'll put it in a centrifuge, and then we centrifuge it for about, say, 10 minutes. At the end of this exercise, the blood in the tube will separate itself into three layers. The basis for this separation into three layers is weight. The heaviest particles of blood will settle first, followed by the next in weight, and then followed by the next in weight. So if you're looking at the tube of centrifuge blood, the particles that you find at the bottom, which means that they settle first, will be the red blood cells. In the middle will be the buffy coats, and at the top will be plasma. Plasma, which is at the top, is composed mostly of water. Water makes up 90% of plasma, and the other 10% is made up of dissolved solutes. In the buffy coat, we have the white blood cells and the platelets. And of course, in the erythrocytes that we have at the bottom layer, those are the red blood cells. Now for the plasma, a main function of the plasma, which is the top layer, will be transportation, because it's made up of mostly water. Plasma also has a regulatory function, regulation, for example, thermoregulation. The buffy coat, which is made up of white blood cells and platelets, its main function is protection. There are two types of protections there. We have protection against disease or infection due to the white blood cells, and then we have protection against excessive bleeding due to the clotting process of platelets. The erythrocytes, which are the bottom layer, their main function is gaseous transport, transportation of oxygen, but also transportation of carbon dioxide. Blood is slightly alkaline, so the pH of blood is 7.4. That makes it slightly basic or slightly alkaline. Now, if you took the same volume of blood and the same volume of water, and you weighed them, the volume of blood, the blood will weigh more than the water. The reason why blood will weigh more than water is because blood has cells and solutes. Pure water does not have cells and does not have solutes. Blood is also more viscous. Viscosity means thickness. So blood is thicker than water. The reason why blood is thicker than water is because, again, blood has cells and blood has solutes. For the volume, males have between 5 to 6 liters of blood, for normal functioning, and females have 4 to 5 liters of blood for normal functioning. If a female has less than 4 liters of blood, she may go into circulatory shock. Also, if a male has less than 5 liters of blood, he may go into circulatory shock. Now, very quickly, let's look at some of the functions of blood. Transportation. Oxygen is transported via blood, hormones, nutrients, the removal of metabolic waste, etc. Another function of blood is regulation. So blood regulates our temperature, blood regulates the pH in our fluids, and blood regulates fluid volume in our tissues. The third function of blood is protection. Blood protects us against excessive blood loss, and that's because we have the platelets in blood, and blood also protects us from infection. That's because we have white blood cells. Plasma, we said earlier on that plasma is made up of mostly water. 90% of plasma is actually water. The rest of the 10%, which are solutes, these solutes, they are mostly proteins. Proteins, they make up 80% of the solutes in blood. The other solutes of blood include nutrients, gases, hormones, waste, electrolytes, etc. The formed elements. Earlier on, we said the composition of blood. We can divide blood practically into formed element and plasma. The formed elements are the living components of blood, and they are made up of the blood cells. The blood cells are the white blood cells, the red blood cells, and the platelets. The red blood cells are technically known as erythrocytes. What is their main function? Gas transport. They transport oxygen and carbon dioxide, but mostly oxygen. How are the red blood cells adapted to their function? Number one, red blood cells are very numerous. They are very abundant. The reason why our blood is red is because of the abundance of red blood cells. So the more red blood cells we have, the more oxygen that will be transported. The second adaptation is, if you look at the structure of the red blood cells, it looks like a donut that is covered on one side. So there is some space in between there. That space is due to the lack of a nucleus and other organelles. Mature red blood cells do not have a nucleus and other essential organelles. So it creates this space for the transportation of more oxygen molecules. The third adaptation is hemoglobin. Red blood cells have a pigment called hemoglobin on their surface. Hemoglobin attaches oxygen onto the red blood cell so that the oxygen can be transported. The reason why the other blood cells cannot transport oxygen or gases in blood is because they do not have hemoglobin to attach the oxygen onto their surface for transportation. Now, the terminology for the formation of blood cells is hematopoiesis. Hematopoiesis means formation of blood cells, and these blood cells are formed in the bone marrow. The blood cells are formed in the bone marrow. That process is called hematopoiesis. Now, for the formation of red blood cells in particular, the process is called erythropoiesis. Erythropoiesis. And the hormone that is responsible, that controls this process, is called erythropoietin. Erythropoietin. Now let's quickly look at some erythrocyte disorders. Erythrocyte disorders. Anemias is one that is characterized by low levels of RBCs. Polycythemia is the next one. Polycythemia is the opposite of anemia. We have an abnormal excess of RBCs. And then we have sickle cell anemia. This is characterized by an abnormal shape of the red blood cells. And then we have iron deficiency anemia, in which the red blood cells, they tend to chain onto each other. They tend to chain onto each other because they do not have enough iron, which is the foundation of the structure of the cell. So one more time, we have 3 erythrocyte disorders. We have anemia, low levels. We have polycythemia, abnormally high levels. And then we have sickle cell anemia. Then we have iron deficiency anemia. If you look at the study material that is provided to you, you should be able to see all of this as they are illustrated. Of course, with anemia, it means that less oxygen is transported throughout the body. With polycythemia, if we have an excess of RBCs, they can actually clog or block small blood vessels. Sickle cell anemia, because they are just a fraction of the red blood cells, they do not transport or they transport very little amount of oxygen. Then, of course, iron deficiency anemia, these red blood cells are also not very efficient. White blood cells. The technical name for white blood cells is leukocytes. White blood cells are the only complete blood cells. In other words, they have a plasma membrane, they have a cytoplasm, they have a nucleus, and they have all of the organelles. So among the 3 blood cells, only white blood cells are complete cells. Their main function is to defend the body against disease or against infection. We can divide white blood cells into 2 groups. We have the granulocytes. They are large and they have a lobed nucleus. Lobed nucleus means the nucleus is divided, but the divisions are not complete, like the lobes of the liver. So they have lobed nucleus. And examples of those will be the neutrophils, the eosinophils, and the basophils. Then we have the second group of white blood cells are the granulocytes. These are small white blood cells and they have a mononucleus. Mononucleus means that their nucleus is not subdivided. These are essentially the cells of the immune system. They are the lymphocytes and the monocytes. Lymphocytes and the monocytes. Leukopoiesis. Leukopoiesis is the formation of white blood cells, and it is regulated by 2 hormones, interleukins, and the colony stimulating factors, CSF. If you want the spellings of these hormones, you could check that in the study materials provided. Leukocyte disorders. We have leukopenia, leukemias, and infectious mononucleosis. Leukopenia, very low white blood cell count. This is a situation that may normally be found in persons that have a compromised immune system. Leukemias, this could be correctly described as white blood cell cancer. So we have white blood cells, but they are not differentiated as normal white blood cells, so they are not able to defend us. Infectious mononucleosis. This is a white blood cell that has been infected by a virus, and when it is infected by a virus, either it will look as if the nucleus of this cell has exploded, or as if the whole cell has actually exploded. Thrombocytes. Thrombocytes, That's the technical name for platelet. So platelet is a common name. The technical name is thrombocytes. They are fragments of large megakaryocytes. So thrombocytes or platelets, they are incomplete cells. They are very essential in the clotting process. Very essential in the clotting process. So they prevent us from excessive bleeding. So whenever bleeding is taking place in any part of the body, then a local mechanism will be activated to stop the bleeding as quickly as possible. That local mechanism is what is called hemostasis. Hemostasis. So hemostasis will be a fast reaction or a fast response, localized response that will actually stop bleeding as quickly as possible. And there are 5 steps to hemostasis, to the process of hemostasis. There are 5 steps. Number 1, vasoconstriction. Number 2, platelet plug formation. Number 3, coagulation. Number 4, clot retraction and repair. Number 5, fibrinolysis. Vasoconstriction, platelet plug formation, coagulation, clot retraction and repair, and fibrinolysis. If you want to get details of that, you could check your notes, but you are expected to be able to know the chronological order or the sequential order in which hemostasis takes place in the human body. Blood transfusion. Blood transfusion. Or before we go to blood transfusion, let's look at some disorders of hemostasis. Disorders of hemostasis. We have the thromboembolic disorders. Thromboembolic disorders. These are cases where we have undesirable clotting. So clotting took place, but it kind of was done a little bit in a sloppy manner. And this could result in blood stasis, that is, can slow down the flow of blood. And you can look at this from maybe an excessive clot was actually formed, and when it does that, it actually reduces the volume. It actually reduces the volume of the vessels through which blood actually flows. So it kind of slows down the flow of blood. The second disorder of hemostasis is bleeding disorders. And these are abnormalities that will prevent normal clot formation. Bleeding disorders. These are abnormalities that will prevent normal clot formation. They could arise from low levels of thrombocytes, or that's low levels of platelets. They could arise from the lack of procoagulants. Procoagulants, these are the chemicals that will help to turn liquid blood into gel-like blood, which is referred to as coagulation, which is very essential for to stop bleeding. Then hemophilia. Hemophilia is a genetic condition that does not allow for easy clot formation. Diabetes too is another condition that does not allow for normal clot formation. Let's turn our attention now to blood transfusion. Whole blood transfusion. Whole blood transfusion means that the person receives blood with all its components. Remember, we'll have the formed component and then we'll have the plasma. So if somebody receives the formed elements and plasma, it means the person has received whole blood. Now, when is whole blood transfusion necessary? When the person has lost lots of blood or substantial amount of blood. When we are treating for the lack of plasma proteins that brings about clotting, and in cases of sickle cell anemia. Cases of sickle cell anemia, large amount of blood has been lost, and when we are treating for lack of plasma proteins that are required for protein. Now, blood transfusion can lead to a reaction. So if the person receiving the blood did not get the right type of blood, it can lead to a reaction. But what is the nature of this reaction? The recipient's antibodies or agglutinogens, will attack any cells that it does not recognize, that it perceives as alien or foreign. It will attack those cells. And precisely what does it do? It destroys the plasma membrane of those cells. It destroys the plasma membrane of those cells. That is what is called hemolysis. It destroys the plasma membrane. Of course, if you destroy the plasma membrane of any cell, then you've essentially destroyed that cell. Now, this brings us to blood groups. Blood groups. The determination of blood groups is called blood typing. The determination of blood group is called blood typing. Now, there are a couple of things that we need to know before we can understand how blood groups, how they come about. The first thing is agglutinogens or antigens. Agglutinogens or antigens. These chemicals are found on the surface of RBCs. These chemicals, we can refer to them as surface chemical markers. They are used to determine blood type or blood groups. Next, agglutinogens or antibodies. They are found in the plasma. Remember the agglutinogens or antigens are found on the surface of the RBCs. The agglutinins, which are the antibodies, are found in the plasma. They protect us from foreign blood. Now, in our blood, we have three types of antigens or agglutinogens. We have antigens A, antigens B, and antigens D. A, B, and D. In our blood also, we have three types of antibodies or agglutinins. Antibody A, antibody B, antibody D, or antibody RH. We can call antibody D as antibody RH. We'll talk about that in a little bit. Now, if you have, so each of these antigens has a corresponding antibody. Each of these antigen has a corresponding antibody. So, for example, if you have antigen A on the surface of your RBC, you're going to have antibody B. So which means that it will protect you from blood group B. If you have antigen B on the surface of your RBC, you're going to have antibody A. If you have antigen D on the surface of your RBC, you're going to have antibody D or antibody RH. We'll explain that a little bit later. Now, let's see how we can determine blood groups. If on the surface of your RBC you have antigen A, then you have blood group A, you are going to have antigen B, you are going to have antibody B in your blood. If on the surface of your RBC you have antigen A, then you'll be blood group A, and then you're going to have antibody B. If on the surface of your RBC you have antigen B, then you have blood group B, you're going to have antibody A. If on the surface of your RBC you have antigens A and antigens B, you are blood group AB, you will not have any antibodies. The reason why you will not have any antibody is if you have, let's say, antibody A, you'll be attacking yourself. If you have antibody B, you'll be attacking yourself. So you do not have any antibodies. Now, the fourth blood group is a situation where you do not have any antigens on your RBCs. If you do not have any antigens on your RBCs, your blood group O. If you have blood group O, you have antibodies A and B in your blood. You have antibodies A and B in your blood. Now let's go back to blood group AB. Because blood group AB do not have any antibodies, they can receive blood from anybody. So they are called universal recipients. They do not have antibodies, so they can receive blood from anybody. Blood group O, they are called the universal donor. They can give blood to all groups. The reason why they can give blood to all groups is because they do not have antigens. Remember, antibodies, they attack antigens. So if you do not have antigens, antibodies have nothing to attack. So blood group O are capable of giving blood to every person. Then let's come back and talk about antigen D, which is called the Rh factor. Rh, it was first discovered in the rhesus monkey. So antigen D, sometimes called antigen Rh. Now, there are two scenarios. It's either you have antigen D on the surface of your RBC or you do not have. If you have antigen D on the surface of your RBC, then you have a plus. Then you are said to be a plus. If you do not have, then you are marked as minus. So if you say you are blood group A minus, it means that you do not have the antigen D. If you say you are blood group B positive, it means that you have the antigen D. So a positive means the antigen D is there. A negative means the antigen D is not there. Now, why is it necessary that we screen blood before transfusion? The reason why we need to screen blood before transfusion is we are checking for compatibility and the presence of disease. So we need to screen blood because we are checking for compatibility and the presence of disease.